Hello students and viewer. So in my first lecture we have seen about basic things which are required for nitrogen fixation. We have seen about abiotic and biotic nitrogen fixation and in which plant which kind of bacteria is isolated for the first time, that we have learned in first chapter. Now this is a second lecture which is based on non-symbiotic nitrogen fixation. So here in this lecture, I'm going to cover all these points. See the points which are I have highlighted in a red color. Non-symbiotic nitrogen fixation, diazotrophs, their ecology, special features of diazotrophs, and enzyme nitrogenase and hydrogenase. So these 6 points I'm going to cover in this lecture. So now see diazotrophs. They are divided into two types. The first type is bacteria and the another one is cyanobacteria. Cyanobacteria, that is nothing but blue-green algae. So free living diazotrophs. Diazotrophs is nothing but what? These are the microorganisms which is going to fix the atmospheric nitrogen. So these microorganisms, they are called as diazotrophs. So diazotrophs, they are divided into two, bacteria and second type is cyanobacteria, or it is called as BGA, blue green algae. Now, according to the mode of nutrition, bacteria again divided into a four types. So which are those? First is aerobic bacteria. Aerobic bacteria is nothing but what? The bacteria which required oxygen for its life cycle or for its survival. So such bacteria, they are called as aerobic bacteria. Second type, facultative anaerobic bacteria. So here the bacteria which is survive in presence or absence of oxygen. So that's why the term comes facultative. Facultative means what? The bacteria which can survive, which can complete their life cycle in presence or absence of oxygen. Hence it is called as facultative anaerobic bacteria. Now the third type is anaerobic bacteria, which means bacteria which completely require oxygen free condition, means in absence of oxygen this bacteria survive. So without oxygen the bacteria will survive or complete their life cycle, they are called as anaerobic bacteria. And the last type, that is photosynthetic bacteria, means bacteria which carried out photosynthesis, they called as photosynthetic bacteria. So likewise, according to the mode of nutrition, these bacteria divided into these four types. Now see which are those aerobic bacteria. So here Azonomonas, Azotobacter, then Bijerinkia, Mycobacterium and Methylomonas. See students, please do remember these names of bacteria, because suppose I'm going to further say only the Bijerinkia, directly you have to the things come, yeah, the things which comes to your in your mind is that Bijerinkia, that is aerobic bacteria, and that is one kind of free living diazotrophs. Getting? So likewise, these things please do remember, and this is not only for further explanation of mine. This is very helpful in your net set preparation. So which kind of bacteria and in which group they are categorized. Next, facultative anaerobic bacteria example. See the example of this. So bacillus, we have heard before this bacillus. So bacillus is facultative anaerobic bacteria. Then enterobacter as well as Klebsiella. So these three, they are facultative anaerobic bacteria. Anaerobic are two. So here Clostridium and D. sulfovibrio. So these two are anaerobic bacteria, whereas photosynthetic are four. So these are rhodomicrobium. See, for photosynthetic bacteria, you might have see this prefix like rhodo and chromo or chloro. Getting? So in which light intensity they are growing, they are surviving. So here it is rhodo, chromo and chloro, right? So you can remember this is the photosynthetic bacteria. So rhodomicrobium, rhodopseudomonas, chromatium and chlorobium. These four are photosynthetic bacteria. Now come to cyanobacteria. Cyanobacteria, they are also called as blue-green algae. This primitive microorganism. So cyanobacteria, in presence of these heterocysts, they are divided into two types: heterocystis cyanobacteria and non-heterocystis cyanobacteria. So as I have explained in my first lecture of this nitrogen fixation, heterocyst is nothing but what? This is the bigger cell which is present within their filament, which is thick walled cell and which is somewhat pale colored. So such cell of that filament is called as heterocyst. So here whether heterocyst is present or not, so according to that two types of this cyanobacteria, heterocystis cyanobacteria and non-heterocystis cyanobacteria. So which are those heterocystis cyanobacteria? So here see Anabina, Anabinopsis and Nostoc. These are heterocystis algae, Anabina, Anabinopsis and Nostoc, whereas non-heterocystis are Oscillatoria and Glyocapsa. I have mentioned only the names which is familiar to you. There are many, right? So please do remember at least these. So Oscillatoria and Glyocapsa, they are non-heterocystis algae. Now they come towards next point. Now the next point is our ecology of diazotrophs. So ecology is nothing but what, in which environment they are present. So this, in which environment they are surviving, in which environment they are present. So see the first point, that is first is variety of habitat. Let me take pen. So now see variety of habitat means what? This diazotrophs, they can able to grow in any of the environmental condition. In any of the environmental condition is nothing but what? They can grow in varying degree of nutrients. They can grow in any pH condition. They can grow in high or low oxygen condition. Means any ecological condition they can survive. So which kind of diazotrophs? So here I have mentioned this is photosynthetic nitrogen fixing bacteria. So this is the bacteria which survive in any kind of habitat, and they are divided into these three types. So first type is non-sulfur purple, second is purple sulfur bacteria, and the third type is green bacteria. So all these three types of bacteria, they are going to grow in any degree of nutrient, pH and oxygen. Okay, and hence we are uh, placed them in under a variety of habitat. Come towards the next point. Let me change waterlogging condition. Getting, so what is waterlogging condition? Waterlogging condition is nothing but what? It is a condition where water is present always. So here flooded soil, then ponds, lakes, then rice field. So in this flooded soil, ponds, lakes, rice fields, which kind of diazotrophs they are going to present? So here see I have mentioned non-sulfur purple bacteria. So this non-sulfur purple bacteria, it is present in a flooded soils, ponds, lakes and rice fields. So along with this non-sulfur purple bacteria, the another example is Clostridium acetobutylicum. So this is the best diazotroph which is present in a waterlogging condition. So this Clostridium is one kind of a bacteria which is present in this waterlogging condition. Clostridium acetobutylicum or Clostridium butylicum and Clostridium pasteurianum. So these bacteria, they are present in a waterlogging condition, specifically in waterlogging condition. Now, the next is optimum temperature. Here in this optimum temperature, which bacteria is going to survive? So under 30 degrees Celsius C. So under this condition, azotobacter will comes, or it is going to survive, it going to perform its function as azotobacter. Now next is acidic soil. So in this acidic soil, which kind of again diazotrophs will be there? So Bijerinkia and Derixia. So these two, they are going to present in a acidic soil. So here see, as I said, all the names of this bacteria, they are going to gives you a particular amount of nitrogen in a particular gram of soil. So suppose in case of this waterlogging condition, Clostridium, it is going to gives you 10 raised to 2 to 10 raised to 6 uh, cells per gram of the soil. Okay, so means in such a conditions, these diazotrophs, they are present. The next, well drained paddy and other crop fields. So in this well drained paddy fields, paddy means rice field, and other crop field, which kind of these diazotrophs are present. So see, in case of these heterocysts, so Anabina, Alozyra, Nostoc and Tolipothrix. So all these four kind of algae, they are present in this heterocyst in well-drained paddy field. In non-heterocystous algae, that is nothing but Lingbaia, Oscillatoria and Plectonema. So these non-heterocystous algae, they are also present in this paddy and other crop field. Please do remember the names. Now we have to look special features of diazotrophs. So here these are very important point, I can say, special features of diazotrophs. So which are those special features, and because of which it going to fix the atmospheric nitrogen? So what kind of features are there within these diazotrophs and which helps to fix the atmospheric nitrogen? So see here, special feature of diazotrophs. So first is site of nitrogen fixation. So which is the site for nitrogen fixation? So here this is very important point for nitrogen fixation in case of diazotrophs. So first I have taken in case of cyanobacteria, in case of algae, blue-green algae. So which is the place or the site which fix the nitrogen, atmospheric nitrogen? So here in case of cyanobacteria, heterocyst is the site which is going to fix the atmospheric nitrogen. Which cell? Heterocyst. I have explained what is heterocyst, and even in the image you can see this is the centrally located rounded cell. This is the heterocyst of that particular bacteria, and rest of this right hand side and even in the left hand side you can see, okay, which is in rectangle shape. So here these two, these are a vegetative cell, and this is somewhat oval, which is larger. This is the heterocyst of that particular bacteria. Now here you can see here, I'm going to again highlight this. I will explain first this vegetative cell. See here in this vegetative cell, which is green in color, this is photosystem 1 and photosystem 2. In case of vegetative cell, both the photosystem 1 and photosystem 2, these two are present. Then next, it is also carried out the electron transport chain. Yes, it's going to carry out because both the systems are there, and simultaneously it is going to fix, it is going to do the photosynthesis, and along with it electron transport chain will run. Then it is also carried out the glycolysis, means respiration of within this cell, uh, and citric acid cycle, and finally it going to form the amino acids. So all these things they are there in the vegetative cell. In case of see heterocyst, now see I'm going to highlight now heterocyst cell. So here in this heterocyst cell, let me explain this inner part, then I will come to this O2 barrier. Now see here both these glycolysis and electrotransport chain is there, and when we compare this photosystem in this heterocyst, the photosystem is reduced. Means what here? See here very carefully. This is only has only photosystem 1. It doesn't have photosystem 2. Okay, why? There again the reason behind it. If the photosystem will be there, then there is the uh, evolution of oxygen, and if the oxygen will be there, then there will be a difficulty in fixing the nitrogen. Okay, and therefore here only reduced photosystem or less or reduced photosynthetic system is present in heterocyst cell. So photosystem 1 is only present in heterocyst. Now I have mentioned here two enzyme names, nitrogenase and hydrogenase, right? So we are not going to look the internal reaction of it. Only we are going to see the site or the place of nitrogen fixation. This further entire cycle I am going to cover in actual mechanism of nitrogen fixation. Just only compare this heterocyst cell with the vegetative cell. So come to its outermost layer, that is this is here it is mentioned O2 barrier. Okay, why it is mentioned O2 barrier? Because this this heterocyst, wall of heterocyst, it contains oxygen binding glycoproteins which together with respiratory consumptions maintain the anaerobic conditions, and hence it is called as O2 barrier wall of heterocyst. And this O2 barrier which is very helpful for nitrogen fixation, means if the oxygen will not enter into the heterocyst, then anaerobic condition will maintain within this heterocyst, and as the anaerobic condition will be there, our enzyme nitrogenase will work efficiently. Means to carry out the activity of nitrogenase we must require oxygen free condition. So student, please do remember, nitrogenase will work only in the absence of oxygen, and absence of oxygen will maintain by whom? The glycolipids which are present on this wall of heterocyst. Getting? So here glycolipids are there. I will write only Gl. Glycolipids are there which are present within the wall of this heterocyst, which which inhibits the oxygen to enter into the cell. And apart from it, so whatever this photosystem 1, whatever the oxygen will evolve by this photosystem 1 will be taken up by whom? Hydrogenase. Means even if the oxygen will enter into this cell, this oxygen will taken up by whom? This oxygen will taken up by hydrogenase enzyme, and then this uh, hydrogenase enzyme will form this H2S, means substrate combined with the hydrogen will formed, and then that will be removed out of this heterocyst, and then nitrogenase will carry out its reaction. Means what? Nitrogen combines with the uh, this hydrogen to form amine, ammonia, right? And further this ammonia getting converted into a amino acid, that is the amino acid name written over a glutamate, that is glutamic acid. So please do remember the site of endophixation in cyanobacteria is heterocyst. So apart from this, see, Apart from this cyanobacteria, such as Lyngbya oscillatoria, where the heterocyst is not there, right? So, in those particular Lyngbya oscillatoria plectonema, in these bacteria, the um, N2 also N2 fixation also takes place. So, it is takes place in particular organized cells, and these cells will again have the reduced level of oxygen. Means it's not compulsory that heterocyst only the cell that will helps in the uh, fixation of nitrogen. Now the see the next bacteria, okay, which is the site in a bacteria which going to fix the nitrogen, right? So in case of this cyanobacteria, in case of blue green algae, heterocyst is the site. Again, I am repeating here. And in case of bacteria, see bacteria, this in a leguminous plants, root nodule. Okay, so root nodule is the site where nitrogen is going to fix. Okay, in which kind of plants? In a leguminous plant, root nodule is the site. Root nodule is the place where nitrogen fixation will takes place. In case of non-leguminous plants, see here, in case of non-leguminous plants, there will not be any nodule-like structure. So what is this nodule and non-nodule? This I will explain in later on in my next lecture. But do remember, in case of this leguminous plant, root nodule is the place where nitrogen fixation takes place. In case of non-leguminous plant, actinorrhiza formed by the frankia, this actinorrhiza is the site for the nitrogen fixation in case of non-leguminous plant, right? And in case of lower plant, lichen is the part where the nitrogen fixation takes place. So these are the site, right? Now next, presence of nitrogenase and reductant. So please student concentrate on this main important slide. This very important slide which is important for nitrogen fixation. Okay, so nitrogenase. See here, nitrogenase is the enzyme which is required to fix the atmospheric nitrogen. So this nitrogen N2 is going to convert into NH3. So this reaction is carried out by the enzyme called as nitrogenase, right? So this N2 is getting converted into NH3 ammonia, and this reaction carried out with the help of enzyme nitrogenase. So now concentrate on this particular diagram. So now see, nitrogenase is made up of these two A and B, these two components. So see here, A part, look at the A part. So A part I have written here MOFE protein. So MO is nothing but what? Molybdo. Fe is nothing but ferro. Molybdoferroprotein. This is called as component 1. This is also called as MOFE protein, or this is also called as nitrogenase. Okay, what it is called as? Nitrogenase. This is also called as nitrogenase. Second component C, it is only Fe protein. This is only Fe protein. So this Fe protein, it is also called as component second. It is also called as Fe, ferroprotein, or this is also called as dinitrogenase reductase. Are you getting? So first component it is called as nitrogenase. Second component is dinitrogenase reductase. And finally, this nitrogenase complex will how it is going to made. See at the diagram this. Let me take this laser pointer. So this is what? This is MOFE protein. This is FE protein. And here see, this is the nitrogenase complex. So nitrogenase complex where only one molecule of MOFE, that is one molecule of only component 1, and here two molecules. See at the pointer, this is the two molecules of Fe protein, means 1 as to 2, means 1 means who one? MOFE protein, that is one molecule, and two molecules of Fe protein will present to form nitrogenase complex, or this is called as nitrogenase enzyme. So always do remember component 1 is very largest unit of nitrogenase enzyme. Question may arise, which is the largest unit of nitrogenase enzyme? So here largest unit is component 1 or molybdoferroprotein or nitrogenase is the largest unit of nitrogenase enzyme, whereas the smallest unit is this Fe protein or dinitrogenase reductase. See, I have written over here this large component, it is non-sensitive to cold. Means even if in the cold condition, it will carried out its reaction, but it loses its activity at 0 degree Celsius. See here. So it is going to lose its activity where? In a 0 degree Celsius. Getting means once we in one hand it is we are saying that is non-sensitive to cold, but yes, in a extreme cold, that is at 0 degree Celsius, it will not work. Hmm? Now come to this component 2. It is a smaller unit and it acts as redox site because a reduction reactions will take place within this site, and hence it is called as redox site. Now come to the detail of this. Come to this this particular this is the structure, a detailed structure of this nitrogenase enzyme. Now see here, let me take a red colored highlighter. So this is okay. So this is the larger subunit of this nitrogenase enzyme, whereas let me take another opposite color. So this is a smaller subunit of this nitrogenase. So this is the larger subunit made up of 2 alpha. See here alpha. Let me take again the pointer. So this is alpha subunit. This also alpha subunit, right? So these are the 2 alpha subunits, right? And these are the 2 beta subunits. So MOFE protein made up of 2 alpha and 2 beta subunits, and within these subunits the P cluster is present. Okay, which cluster? P cluster is present. So this P cluster is also called as P center. This is also called as P center, right? And also it has ferromolybdo cofactor, or it's also called as iron molybdenum cofactor, FeMoCO. Getting? So FeMoCO is nothing but what? Iron molybdenum cofactor it also has. Okay, and this cofactor it converts substrate into its product. So this is the detailed structure of larger subunit, that is MOFE protein. Now come towards its a smaller part or smaller subunit, that is Fe protein. It has two gamma subunits and it has now FeS4 cluster. See, In larger subunit, which cluster is present? P cluster is present in a larger subunit, whereas in a smaller subunit, FeS4 cluster, and that cluster is also called as M. This cluster is also called as what? M cluster. M cluster, and this is cluster. P cluster present in MOFE protein, or P cluster present in larger subunit, whereas M cluster or Fe4S4 cluster is present in a smaller subunit. So this is the structure of nitrogenase. So always do remember, as I have explained you before, nitrogenase is the enzyme which is required to convert nitrogen into ammonia, and nitrogenase will perform its activity in the absence of oxygen, right? Oxygen free condition. So nitrogenase conditions are there. If the oxygen will be there, then nitrogenase will not be work, and if the nitrogenase will not be work, our nitrogen fixation will not be taken place. So please remember this presence of nitrogenase enzyme, which is helpful for conversion of N2 into NH3. Now see again, I have given this one particle of this, one molecule of MOFE and two molecules of Fe, right? And then it is going to form our enzyme, that is nitrogenase enzyme, and nitrogenase will carry out the reaction which I have placed likewise. So one molecule of, for the fixation of one molecule of nitrogen, see how much of ATP is required. So 12 ATP and 6 hydrogen with 6 electron they are required, and it will going to form 2 ammonia, and it going to reduce this 12 Mg ATPs into a 12 ADP plus 12 inorganic phosphates. So see students, as the ammonia will formed within our cell, so this ammonia is not again useful in our plants. If it is remain as it is within the plant, that will disturb or that will distract our plant or whole metabolism, whatever the reactions which are going to perform within the plant, that will disturb with the help of ammonia, and hence this ammonia should convert immediately into its uh, rest of the factors like NO2 or NO3. Getting? So ammonia should not be remain as it is within the cell, that ammonia should be removed, or ammonia should be divided into its uh, rest of the part, that is nitrite or nitrate. Now this is next, presence of hydrogenase. Okay, so till here what we have seen, the special features of diazotrophs. The first point we have seen the site of nitrogen fixation. Then second point we have seen presence of nitrogenase, and third point now it is presence of hydrogenase. Now what is the main role of this hydrogenase? So see here, so many diazotrophs evolve hydrogen during nitrogen fixation, which in turn inhibits nitrogen fixation reaction. See, who is going to inhibit this nitrogen fixation? Who is going to inhibit? Hydrogen. And hydrogen, when it is going to evolve, so hydrogen it's evolved during nitrogen fixation. Now so to get protection from inhibition by H2, many diazotrophs possess an enzyme called as hydrogenase. Okay, so hydrogenase, what is the main function of hydrogenase? I have explained earlier. So hydrogenase is the enzyme which is required to fix the hydrogen which is evolved during nitrogen fixation, right? So this hydrogen is reutilized to produce more ATP, and hence it improves the efficiency of nitrogen fixation. Getting? So hydrogenase is the enzyme which going to combine with the hydrogen, right? The which is produced during nitrogen fixation. So for the first time, this utilization of H2 by purple bacteria was discovered by Roelfsen, the scientist in 1935. See the year in which this discovery has been done. So, so first time utilization of hydrogen by this purple bacteria, and with the help of hydrogenase enzyme, this hydrogen is going to combine with the substrate, and this hydrogen going to form hydrogen substrate. So this is the reaction. Now, last part, that is presence of self-regulatory system, right? Means being we have seen these all these conditions, means site of anthropization, then presence of nitrogenase, then presence of hydrogenase in a plant or in a microorganism. Some self-regulatory system should be there to conduct or to carry out nitrogen. So which are those self-regulatory system? So see here in my next slide. So first is azotobacter. So azotobacter, it is the bacteria which carries out nitrogen fixation. So, how the self-regulatory system is present in the azotobacter which helps to fix this atmospheric nitrogen. So, in case of this azotobacter, uh, the protection is given to this bacteria. How? Because this bacteria, they has the high respiratory rate, right? And due to this high respiratory rate, oxygen is prevented by reaching the site of nitrogenase reaction. Okay, so means whatever the oxygen will be there, that will not be reached to the site where nitrogen fixation takes place, and hence azotobacter is going to fix the atmospheric nitrogen. Means this is the self-regulatory system. Which system? High respiratory rate. This is the way through which the O2 is going to compensate and nitrogen fixation will take place. Then next, cyanobacteria. So as I have explained you before, heterocyst is the cell where the nitrogen fixation takes place. But again, which is the self-regulatory system? So here, self-regulatory system in which means what? The absence of photosystem, or if it is present in a reduced form, right? So I have mentioned earlier that in a heterocyst, only photosystem 1 is present. Photosystem 2 is absent in heterocyst, right? So photosystem 1 is present, and hence the nitrogen which is not inhibited by the O2 evolved during photosynthesis. And if it is present, okay, if it is present, again the hydrogenase enzyme will helps to maintain the O2 tension or anaerobic condition, and hence the nitrogenase will work fluently within this heterocyst cell. Now see, come towards this non-heterocystous species. Non-heterocyst species. So here see oscillatoria and lingbia. These are the non-heterocyst algae, and in this algae, such cells means what? The cells which are going to fix nitrogen. So these cells, they are located in the center. See where they are located? They are located within the center of the cell or the colony. So these cells do not produce O2 photosynthetically. See, this is very important characteristics of non-heterocystous species. Okay, they are not going to produce O2, and they have again thick wall. Okay, thick cell wall, and because of this they are going to maintain an aerobic condition, and they are going to fix the atmospheric nitrogen as like in heterocyst of other bacteria, cyanobacteria or blue-green algae. Getting? So in a non-heterocystic species, which self-regulatory system is there? These nitrogen fixing cells, they are present or they are located in the center. First thing. Second thing is they are not going to synthesize or produce oxygen. And the third thing is they has thick cell wall, and because of these three conditions, the nitrogen fixation takes place in non-heterocystous species. And come to the last point, leguminous plants. So leguminous plants, leg hemoglobin, see, this is the very important protein, leg hemoglobin, right? So hemoglobin, which is present in our body. Okay, so hemoglobin, what it does in case of human beings, it is going to combine with the oxygen, right? So here the hemoglobin which is present in a leguminous plants, they are called as leg hemoglobin. So this leg hemoglobin within the leguminous plants, they are going to regulate the oxygen level within this, and hence the nitrogen fixation will take place. Getting? So here recall yourself just four special features what we learned, that is site of endophixation. Then second point is presence of nitrogenase and reductants. Then third thing, presence of hydrogenase and presence of self-regulatory system. So likewise, in four different conditions, I have given the four different self-regulatory systems. So here you have to remember these points, and even if you have any query, please ask me. But please do remember the nitrogenase and hydrogenase. Nitrogenase required for endophixation and hydrogenase required to maintain the O2 tension within that particular cell. So thank you very much for patience listening.